All right, so today's video is going to be about epistasis, is where we're going to start, and then a couple of other little scenarios and information about some disorders. Okay, so first thing, epistasis is a situation where one gene, controls the expression of another gene. All right, so in simple terms, there's going to be one gene that has to get turned on in order for a second gene to actually work. And so there's a lot of scenarios like this, but our most common is going to be lab coat color. Now, when I'm talking about labs, I'm talking about the dog. It's a Labrador Retriever. And the color of the lab is based on a situation with epistasis. So black is dominant to brown. And so lots of times with brown labs, they call them chocolate. And labs are a very big business and people make a lot of money breeding labs. But there's another common color. Now there's all kind of new genetic traits that they've bred and different things. But your three basic colors are going to be black, brown, and then we have yellow. Okay? And yellow is basically going to be almost kind of like an albino version of the dog. And the reason is because there's actually two genes involved in this dog getting its coat color. So gene number one turns the color on or off. Okay, so, and then gene number two determines the color. All right, so for example, if you were big B, and this gene was on, that lab would be black. Big B, little b, and that gene is on, also it'll be black because black is dominant to brown. If the gene number one gets turned on, but that dog gets two little b's, it'll be brown or chocolate. Now, yellow, if you've noticed, there's not an allele for the yellow trait. That's going to be when this gene is off. Okay, so the off version, it doesn't even matter what the second gene is because it doesn't even get to turn on. So when gene one gets turned off, this is how we're going to get yellow labs. Okay, so I'm going to show you how it works in a Punnett square. So we'll call, we'll use big E is on and little e is off. Okay, and that would be for making the color or not. And then big B will be black and little b will be brown. Okay, so we're going to cross two black labs. So in order to be black, they have to have at least one big E, and then they have to have at least one big B. All right, because the other letter doesn't matter. The capital E is always dominant. So I'm going to make them both heterozygous. So I'll make it big E, little e, times big B, little b, and then big E, big B, little b. Okay, so both of these labs are black. So this is a black lab times a black lab. All right, so when we do our Punnett square, we're going to do our same deal. Draw our box, and we're going to FOIL our letters. So big E, big B, big E, little b, little e, big B, little e, little b. These are the combinations of alleles that parent number one can give to the offspring. Second parent is going to be the same set of combinations. So he can give the on gene with the black allele, the on allele with the brown, off with the black, and off with the brown. Okay, so in this situation, we'll fill in the box. So two E's, two B's, E, and then big B, little b, and then E, and then big B, oops, big B, little b, big B, little a. Okay, so in this situation, our phenotypes, we only have three. We can have black, we can have brown, or we can have yellow. Okay, and then our ratios are going to be out of 16 because we have 16 boxes here. All right, so in order for them to be black or brown, they have to receive the on gene. Okay, so I'm going to go ahead and do all the ones that did not get the on gene. And so this one is off because there's no big E. This one is off, this one is off, and this one is off. Everybody else gets to have color, but these four options, no matter what their letters are always going to be yellow. So 4 out of 16 chance that there is a yellow lab between these two black labs. All right, and then this one's going to be black, 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 and then black. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of 16. And then that one's brown, brown, and brown. So 3 out of 16. And double check 9, 3, and 4 equals 16. So in this cross between the black lab and the black lab that are both heterozygous, you're going to get 9 out of 16 chance for black labs, 3 out of 16 chance for brown, and 4 out of 16 chance for yellow. All right, so I want you to do 2 crosses and then pause the video and then come back and see the answers, okay? So the first cross I want you to do is a yellow lab times a yellow lab. And then I want you to do that lab. These are 2 different crosses. So this could be number 1, number 2, and then we'll do, oops, that lab. Okay, so I want you to tell me what the offspring will be here and what the offspring will be here. Okay, so with the yellow times the yellow, yellow labs don't have any of the genes that turn the color machine on, so they can't make any pigment at all. So it doesn't even matter what the B's are for them at that gene because E has them off and has them off. And all of their offspring will be yellow. So 2 yellow labs will give you 100% yellow baby labs. So if you're selling yellow labs, you want to cross breed 2 yellow labs. Now over here, this lab has the gene for color, but this lab doesn't. Okay, so this one's definitely going to be yellow. And over here, this one has the gene for color, but it'll be brown. But when you cross them, you're going to have a brown or chocolate lab with a yellow lab. And this parent can only give the on gene and the brown gene. And this parent can only give the off gene and the black, or excuse me, the off allele and the black allele. So in the box, it'd be big E, little e. So they'll be on, so they get to make color. And then it'll be big B, little b. And so they won't look like either parent. It will be 100% black labs. Okay, so when you do epistasis, there's 2 genes involved. One gene turns it on, and then the other gene determines the color. So this is what happens in albinism for humans. They have to have the gene that turns the melanin production on, and then they can make their skin color. So people who have albinism, actually, it's a situation of epistasis. So they have the genes for skin color, eye color, and hair color, but they inherited the off gene, and so therefore their body cannot make those colors. So they can have babies with normal hair color, skin color, and eye color, but they don't have it. All right, so our next little scenario I want to talk about is polygenic inheritance. So polygenic inheritance. Okay, so poly means many, and then genic means genes. So this is when many genes add together to give you an actual phenotype. So the phenotype is like this gene plus this gene plus this gene plus this gene, and then you get the actual phenotype. Okay, so skin color is one of those examples. And skin color in humans can be controlled by 3 or 4 different genes. So you get 2 letters for every gene, one for mom, one for dad. And we're going to make capital D for dark and lowercase d for light. And so depends on, so you could get 2 dark genes here, 2 light genes here, a dark and a light, and a dark and a light. So this child gets 1, 2, 3, 4 dark genes and 1, 2, 3, 4 light genes. So their skin color actually becomes a blend. So it's a blend of 4 darks and 4 lights. And we did this when we did our genetics babies. So in your book, this shows it when it's a combination of 6 alleles, which would be 3 genes. So if someone inherited 0 dark genes, then they would be the lightest. 1 dark gene, 1 dark allele, 2 dark alleles, 3 dark alleles, et cetera, et cetera. So this one just shows it an additive effect when it's on 3 genes, because there's 6 alleles. So you always divide it by 2. All right, so height can sometimes be like that. So if we did tall is dominant to short, and there's gene 1, gene 2, gene 3, gene 4, and gene 5. Okay, so if there's 5 genes, how many alleles are we dealing with? It's double. So 5 genes equals 10 alleles. So 10 alleles. So let's say they get this, all right? So in this scenario, they only got 2 tall genes and the rest are short. So 2 tall and 8 short. So just like we did the activity with our little babies, our fake babies, this would show there wouldn't be a whole lot of height. So you start with the base height and then you would just add 2 tall to it. So in the genetics questions, sometimes they'll do something like this. So they'll say like plant base height of 5 centimeters, okay? And then each dominant allele adds, let's say, 1 centimeter of height, okay? So if you have this parent, and we'll say little a, little b, little e, and they cross with the big A, big B, big B, big E, okay? Now, each dominant allele adds a centimeter of height, but the base height is 5. So this parent plant has no dominant alleles. So their base height is going to be 5 centimeters because they can't add anything to it. This plant gets to add a centimeter for every dominant allele. So they get to go start at 5, and they get to add 1, 2, 3, 4, 5, 6. So they get to add 6 centimeters. So basically it's a 5 centimeter times an 11 centimeter. Okay, now we're not doing math. This is their height. So that's a short plant and that's a tall plant. Okay, so that's the shortest version and the tallest version. When you cross these two, this is like a P generation, pure recessive, pure dominant. So all the offspring will be heterozygous. So based on this cross, every single one of the offspring will be how tall? Take your base height of 5 centimeters plus the number of dominant alleles. So this one has 1, 2, 3. So they add 3 centimeters of height. So that plant would be 8 centimeters. So all the offspring would be 8 centimeters tall. All right, so let's do this cross real quick and then pause it, do the cross or answer the question, and then turn it back on and see if you're right. So tell me how tall each one of them is. Oops. Okay, so in this situation, we started base height of 5, and then every dominant allele, 5, 6, 7, 8, 9. So that one's a 9 centimeter tall plant. This one's 5, 6, 7 centimeter tall plant. And then this one's 5, 6, 7, 8. So in these, it doesn't really matter where your dominant allele is. Was it gene A, gene B, or gene E? It's just an additive effect. So it's going to be a polygenic trait. And so for all the dominant alleles that you have, you just keep adding to it. So depending on, the problem or whatever, it could be a plant height, it could be the length of a tail, it could be the size of an organism, it could be the height of a human. You're just going to keep adding to a base number for every dominant allele that they have. Okay, the last thing I'm going to show you is when you get a crazy Punnett square, what you think is going to be crazy, and you can do the shortcut. All right, so what if I told you I wanted you to do big A, little a, big B, little b, big E, little e, big F, big G, little g, times. Okay, you might cry because that literally might be more than you ever wanted to do. But when you see one of these, they're going to ask you to, it's kind of like a divide and conquer type situation. So the question would be something like, what's the chance of getting this offspring from these two parents? Okay, so what's the chance of getting that? All right, so what I want you to do is I want you to take each set of letters and do a separate Punnett square for them. So you would do the big A little a times big A little a, right? And in it you have big A, big A, big A little a, big A little a, little a, little a. And what I want you to do is I want you to pause the video and I want you to do it for each set of letters that I have here and then come back. Okay, so it's a little bit sloppy, but basically what I did is I did my Punnett square for the A's, the B's, the E's, the F's, and the G's, okay? So now I can separate it and look at them individually and figure out the chance of each combination. So for 2 little A's, I would look at this Punnett square, and the chance of 2 little A's in that Punnett square is 1 out of 4 because it's only in that spot right there. Big B, little b. I look at the B Punnett square. Big B, little b actually has 2 out of 4 chance. Big E, little e. I go to this box, and there's actually 2 out of 4 chance there. 2 little F's. Go to the F box, and it's only 1 out of 4 chance. And then 2 little G's, 1 in 4 chance. So I do each set of each gene and their alleles separately. And then to get the possibility of the whole combination, I multiply them all together. So now I have a fourth times 2 fourths times 2 fourths times 1 fourth times 1 fourth. So you do all the tops across. So all the way across, 1 times 2 times 2 times 1 times 1 is 4. All right, and then 4 to the 5th power. I have to use my calculator here. I think it's 256, but 4 to the 5th, no, it's 1,024. All right, so the chance and the probability of this parent times this parent getting this offspring is this possibility. Okay, so I'm going to give you a couple more options using this same cross, and I want you to give me the probability of getting these offspring. So the first one is big A, little b, little e, little e, big F, and two big G's. So I want you to tell me what's the chance of getting that offspring from these parents. And then the second one I want is big A, big B, big E, little F, little G. Okay? Now, little twist, I want you to tell me the chance that they phenotypically look like their parent. Okay? And that's a little bit of a trick. So I want you to pause the video. I want you to answer the first one, the second one, the third one, and then come back and see what the answers are. All right, so for big A, little a, it's 2 out of 4 chance. That's 1 out of 4. That's 1 out of 4. That's, 2 out of 4. And then that's 1 out of 4. So when you multiply across the top, once again, it's 4 out of 10,024. You can probably simplify that a little bit more. Let's see, 10, 24. So that's 1 out of 256 chance. So 1 out of 256 chance on both of those. Not a big chance, okay? Here, the chance here is 2 out of 4. Here, the chance here is 2 out of 4. 2 out of 4, 1 out of 4, and 1 out of 4. And once again, I'm just looking at my Punnett squares that I did separately up here. I'm separating them. And so this one's actually going to be 8 out of 1,024. And so that will simplify down into 1 out of 128 chance. Okay, so not a terribly big chance, but there's still a chance. Okay, now, phenotypically looking like their parent. All that means is the parent has the dominant phenotype for every trait. So the child only has to have at least one big A, and then it has to have at least one big B, one big E, one big F, and one big G. Because the second letter doesn't matter, because then they'll physically look like the parent, because the parent has the dominant A phenotype, dominant B phenotype, dominant E, F, and G. Okay, so in this situation, the chance of having at least one big A is 3/4. The chance of a big B is also 3/4. This is 3/4, this is 3/4, and this is 3/4. So then on the bottom, multiply 4, 4, 4, 4, 4, we get 1024. Now it's going to be 3 times 3 times 3 times 3 times 3, and it's going to be 243. And I don't think that simplifies, but we'll leave it at that. So it's 243 out of 1024. So that is the chance that they phenotypically look like the parent. And then exactly like the parent. They have all the traits just like the parent. All right, so that's how you do those, and that is it. I hope that that was helpful, and I'm going to give you some practice problems, and just make sure you keep your notes.